Thank you very much for coming today and joining us for this session. My name is Sandra Diaz. I am the scientific lead of the Simulation and Data Lab Neuroscience at the Jülich Supercomputing Center. And I am delighted today to share this webinar instance, which has a different format. So today we want to share a bit about the role of high-performance computing in neuroscience. And we have a set of short use case talks. But first, I would like to give a brief introduction. Then we will have the presentations, and then we will have conclusions and questions and answers. So yeah, I will dive directly into the topic. And of course, you all know that today neuroscience really addresses questions that touch many different scales regarding the brain, from the molecular level all the way to the whole brain. And we are collecting large amounts of multimodal data, from experimental data, clinical data, genetic data, about the structure as well as the function of the brain. And together with all of that, we are also getting an increasingly and more available computational power, and that comes together with new methods to leverage that computational power and to develop new technologies. So today I really want to invite you to think about the role that sometimes supercomputing has had in the development of neuroscience research as a whole, which sometimes is a bit hidden behind as an enabler, but not so much on the forefront. And in the case of e-brains, this has been a really visible thing. Since the Human Brain Project started, high-performance computing played a very critical role. And I just want to go through some highlights that have been around and enabled by this collaboration between neuroscience and high-performance computing and advances in technology, to make this relationship a little bit more visible to all of you. So, of course, we can today understand the structure of the brain far better. We have access to large amounts of data that allow us to understand the cytoarchitectonic structure of the brain to unprecedented levels of detail. And of course, this is thanks to the combination of the vast brain atlases that there are around now, together with the large computing infrastructure behind that allows to treat that data interactively, and also to explore it, analyze it, and to look at different features of the data and connect that to mathematical models. Of course, then we have high performance computing is also behind the simulation of the brain at all scales. Of course, the more detailed, the higher computational load. For example, in the molecular and subcellular level, the amount of computation required is enormous. And we are seeing now more and more need of novel technologies in the back, like quantum computing, neuromorphic computing, being able to support and push these very strong loads of computation. But this integrated view of the brain and creating models and simulation all around is, of course, more and more possible and feasible with the help of high-performance computing. We, of course, can have now large amounts of data integrated into extremely detailed models, like these examples from our colleagues who have developed basal ganglia models, models of the hippocampus, models of the cerebellum. All of this done in the context of the Human Brain Project and e-brains, and which have allowed to get insight on the dynamics and the relationship between structure and function of the brain, really at a new scale. And of course, also visualizing the outputs of those simulations, which then high performance computing allows to then get all this data and put it together into a reasonable way to understand it and navigate through it. Yet again, we also need to create models that are robust and that do not only cover a small area of our hypothesis, but that are really applicable to many different areas and many different features of the brain dynamics. So high performance computing also helps us observe and explore the parameter space of the models that we have created based on the data, based on the structure, and on our own insight of how things work. And this can be done in an interactive process, of course, exchanging information with simulations, for example, with running Nest, Neuron, Arbor, the virtual brain, the brain scaffold builder, SNUDA, Mimic. We have so many tools that have been developed in the context of the Human Brain Project and eBrains, and that have led us to a very powerful set that we can leverage for this. High performance computing is also there enabling the new generation of neuroscientists to get interact with new models of the brain through these wonderful tools like Nest Desktop that allow researchers to explain in a very intuitive and graphical way how to create models and how to execute them, and creating an environment where training and teaching is also possible through the combination of cloud infrastructure and HPC infrastructure. And of course, HPC is complemented by this new ways of architectures like neuromorphic computing, which in the case of eBrains is highlighted by the BrainScales and Spinnaker technology that enable new ways of thinking about compute and to challenge the limits of computing that we have with traditional architectures. Finally, I also want to share the experience of the community using high-performance computing. We have this project, the Virtual Brain Twin, which is also partner with eBrains and which has the goal of creating a virtual brain twin for the treatment of schizophrenia. And in this case, we are using combination of multi-scale modeling, machine learning, high-performance computing, and big data to create these new services to understand the brain and then personalize the treatment of patients who have a very difficult time dealing with the traditional ways in which these procedures are normally implemented in medicine. But in this case, the HPC systems that we are using have not only been an enabler of computing, they have also become a virtual environment where the colleagues meet and we co-design methods, we share data, we implement code, we do our everyday science in there, and we advance science together. So I want to also transmit that high-performance computing today is not only a back-end tool, but also a space where we can create new science. And with that, I want to close this introduction. I hope that I have given you a broad overview of just a few of the so many applications that have been boosted by high-performance computing in the past. And now I want to give the floor to my colleagues who will present their use cases and who will show us also a path forward in the application of high-performance computing into new questions in neuroscience. So first, I would like to ask my colleague Christian Schiffer, who is working at the Forschungszentrum Jülich, to share with us his work towards the creation of a foundation model of the brain. Christine, please.
Yeah, thank you. Let me
Take the floor.
quickly share my screen. So I would like to apologize in advance if I'm coughing in between. I'm just recovering from a cold, but I hope it's not too bad. Yeah, good morning, everyone. Thank you for the nice introduction. My name is Christian. I work at the Institute of Neuroscience and Medicine, INM1, at Forschungszentrum Jülich, where I lead the team Large-Scale AI for Brain Mapping. And today I want to talk about our efforts towards a foundation model for the human brain. And in this talk, this will partly be based on on work that we have already done, which has just been submitted for journal publication, but also a little bit outlooking to show you a little bit of what we are planning to do and how HPC plays into this into these plans. So one of the core goals of our lab is to develop AI methods to describe human brain architecture, so the structure of the brain in general, with a particular focus on micrometer resolutions and at the whole brain scale. Which includes, for example, the organization of cells, fibers, and perspectively also function. So our goal is to develop methods which we can basically give data. These methods then encode the data in some useful representations, which we can then use for certain kinds of analysis, which I will show some examples later. The way we approach this in our institute is that we look at post-mortem human brains. We take these brains, fixate them, cut them, which gives us thousands of histological slices per brain, which we then, depending on what we want to look at, stain and then digitize, which gives us high-resolution images showing us different organizational aspects of the brain. Some examples, very prominent examples, include, for example, cell body staining, giving us insight into the cytoarchitecture, so the cellular architecture of the brain, polarized light imaging, giving us these beautiful images showing us the fiber architecture of the brain, and also receptor autoradiography, giving us insight into the chemoarchitectural organization of the brain. So our goal is to build models which can take, in the best case, all these modalities and maybe even more, and encode them in a ways to make them available for analysis. In our initial work, we have focused a lot on cytoarchitecture because it's widely available. It has a rich history, and there's a lot of data available. If for those of you who are not familiar with this, cytoarchitecture is defined by the organization of cells. For example, here in the cerebral cortex, where we can see different organizational principles, for example, the organization into layers, and also depending on where we are in the brain, we have different composition of these layers, for example, in terms of cell size, cell density, and so on. Like here in the motor cortex, where we have these giant beta cells. And the interesting part here is that by looking at how the architecture looks like, so essentially looking at the hardware that we run on, we can also better understand how the brain functions and how it behaves in health and disease. So what we want to do with this data is we want to encode brain architecture. We basically want to build a model, as I said, to which we can give the data, and then this allows us to answer certain questions about the data. For example, what kind of cells are present? For example, these beta giant cells. How are cells distributed? How are layers composed? Which areas are formed? And then for other modalities, also questions like how are fibers organized and how do local networks function? Of course, we could build AI models to solve each of these tasks individually, but this is not really efficient because in many cases it's a little bit fuzzy and we don't have much training data, for example, to achieve this. And the more modern way to achieve such goals is to build so-called foundation models. Foundation models have been a revolution in the AI field in the past years, with models like most prominent one is probably ChatGPT. So models which are trained on broad amounts of data, not specialized for a specific task, but very, very adaptable because of the fact that they are able to represent the data that they are operating on in a very, very efficient way. So to summarize, our goal is to build such a foundation model, which is in first place able to work on cytoarchitecture, but method-wise, this is also applicable, hopefully later also to other modalities. So the question is, okay, we want to build a foundation model, but how do we learn? So like, how do we actually train this thing? We all know that AI is only as good as the data and also the objective that we use for training these models. And what we use here is, to go a little bit into the method details, is we use contrastive learning from spatial proximity. Contrastive learning means that we want to make the model map samples, so in our case image patches, which are in some sense similar to similar vector representations, because this, if you think about it, for example, allows us to later differentiate, for example, brain areas, because they are, if they are deemed similar, they are mapped to similar vector representations. To give an example how this looks like. So assume we have two brain sections from two different brains, but from two approximately similar locations. This is somewhere in the frontal part of the brain. And we assume that we have different locations within this brain. Now we can see that our assumption is that the closer something is in the brain, the more similar it is. Of course, probably many of you will say, okay, this is not true in many cases, but it's good enough as a proxy, and this is all that we need for our AI training. So what we say here is that the orange and the blue one, they are very close to each other, even though they are in different brains, but the purple one is relatively far away. So we basically train our model to map the orange and the blue patch to a similar vector representation and the purple one to a different one. There's a technical difficulty that we can't really measure distance between different brains so easily. So we don't do this actually in the image space, but we do this in a reference space, which is something that is quite easy to do. And now we have everything that we need to train our AI model. We can extract high resolution image patches from our microscopy data. We can then pass them through our neural network model, get some vector representations, and now we say, based on the distances that we computed in our reference space, we want to make certain representations similar and certain representation dissimilar. We do this using a specifically designed objective function, which I don't want to go into too much detail here, but the point is that it is an adaptation of a already existing learning function, which we basically adapt to work with. So this is the info NCE loss, which we basically adapt to work with our idea of having spatially close samples encoded in a similar way. So this is the training objective that we define. The question is now, like, how do we actually train this? And this is where HPC comes into the equation. So we train these on HPC, otherwise I wouldn't be talking here, probably. And the data that we use is 10 brains from our in-house brain collection, which is not the entire collection, but this is what we started with, which was about 4,500 sections. The data volume is about 11 terabyte, and we sample 1 million image patches for training at 2 micrometer resolution. And here it's important to note that even the image patches that we use, so the one that you see at the bottom, are in themselves very, very large. They are about 2,000 by 2,000 pixels, which is, for those of you who are familiar with like image classification AI models, for example, which is quite a lot. Usually people sample patches from this size of images. And we assume that for each patch we have a corresponding coordinate, in our case in the colon 27 reference space. The architecture that we use is a slightly modified ResNet 50 vision transformer hybrid, and the current compute that we require, so this is basically our standard setting, includes 64 A100 GPUs, which takes about 28 hours depending on the training that we are doing, which we at the moment do on the Eureka DC system at the Jülich Supercomputing Center. So this is our status quo. I will, at the very end, talk a little bit about how this will scale up in the future. So after training this, what can we actually do with this model? I talked about the technology and the method, but what can it actually do? This is probably the most interesting part. So the point is that after training this thing, we have one model that we can use for many applications. This pre-training is very, very generic. It gives us representations, but what can we actually do? One prominent application is that we can use it to automatically classify brain areas. We have just seen in Sandra's presentations the Jülich Brain Atlas, which has been created by manually annotating brain areas over the last decades. And we can use this data actually to fine-tune our foundation models for brain area classification. And this is actually also something that flows back into the eBrains infrastructure, for example, by creating very, very detailed maps of thousands of sections in the big brain data set. Similarly, We also can use it for layer segmentation. So if we're not interested in the brain area per se, but the actual composition of the layers within these brain areas, we can also do this. So if we have a relatively small amount of data, training data, actually, so we went down to as low as 10 annotated training patches, and it still works reasonably well. We can use this model to do layer segmentations. But there are also a little bit more exotic applications. For example, we found that we can use this quite well to quantify the result of generative models. So if we have other AI models which generate data, there's always the question like, is this plausible? Is this something that we can use? And we can actually use this model to quantify how well this data is. How well this data looks like, basically. We also found that we can use the features for data-driven discovery of new brain structures. So if we look at the few remaining white spots in the brain where we actually don't know which brain area should be there, we can actually use clustering on the features to find new areas, which is something that is super interesting. And because people always like that, if we have also something interpretable and not just an abstract vector representation of a model, we also found that we can also go basically close the loop and bring these features back to something that is interpretable. So, for example, cortical thickness, or the thickness of individual layers, or the density in individual layers, which is something that is very nice. We can also go a little bit even more abstract in looking at how does this feature space that the model learns actually look like. This I can't go into too much detail here, even though this is more or less my favorite part. But basically, we can look at how the model sees the data using this kind of embedding representations, and then learn something about how the brain is actually structured and how, for example, it varies across the brain. So, for example, what we see here is that we get these clusters, which are brain clusters, which the model is not trained to do, and it still does it, which is quite interesting, I think. This brings me already to the end, and I promise that I will give a little bit of outlook also in terms of scaling up. So we have several axes of where we want to basically scale this current approach up in the future. One obvious one is data. So we have been focusing a lot on cytoarchitecture so far. We are now in the process of extending this to other modalities, for example, PLI data, but also we're looking at MRI data, HIP-CT data, which brings us from the range of terabytes to petabytes, which of course is computationally a quite big challenge. We're also going to scale up the models themselves, so in terms of model parameters, because we found that in general, larger models perform better. This is the case in other domains as well, and for us, it seems also to be true. So we're looking into other model architectures, and we're also aiming to scale up a little bit in resolution from 2 micron to 1 micron, which doesn't seem a lot, but from an application perspective, it's quite important, because, for example, if you go to 1 micron, you can suddenly use this model for things like cell detection, which it can't do really well at the moment. And technically, it's also quite a big challenge, because the difference between 2 micron and 1 micron is the difference between barely fitting on an A100 and not even fitting on an H100. So that's quite a challenge. To make this work, actually, we are very, very happy that we are one of the pioneer projects that already have access to the new Jupyter supercomputer at JSC, where we are currently seeing how large can we push our AI workflows. And we are quite proud that we are already able to scale this to about 4,000 compute nodes. Not very efficiently though, but it runs, which is quite cool. And yeah, we're now looking forward into how well we make this work on such a big machine. This brings me to the end of my talk. I would like to thank everybody who contributed to this project in some way, and of course you for your attention. And I'm happy to take questions now or at the end.
Thank you very much for the very nice presentation, Christian. That was really nice.
Thank
I
you.
think we will leave the questions all to the end.
Yeah.
So then I would ask David if you can take the floor. My colleague David Kappel, who's professor at the University of Bielefeld, and who will share with us about his current work at the interface between neuroscience, artificial intelligence, and many interesting the topics developing at high speed. Thank you very much, David, for joining us today. I leave the floor to you.
Yeah, thank you, Sandra, for the introduction and the invitation. I'm going to give a brief overview of what we are doing in my group in Bielefeld. So let me see if this works. Yeah. So what we are doing, we are come from a quite different direction, but I guess we use very similar methods in the end. So the idea of our research is to actually take state-of-the-art machine learning models and also incorporate insights from neuroscience and transfer from what we have currently to more efficient machine learning. So basically, what we can learn from the efficiency that we observe in biological brains. And I just pulled out two examples in this talk, which is one. So basically, we identify principles in the brain that enable efficiency, and then we transfer them to machine learning models. And one of these principles we have been looking into quite deeply is event-based processing. As probably many of you know, the brain operates with events. So these are these action potentials that you see here. This is a very nice simulation already from a couple of years back, where you see this action potential formed in the soma of a cell. And this is a very generic principle that can be transferred into modern machine learning models and to improve the efficiency, as I will show. And already a couple of years back, we had developed an algorithm that essentially takes an existing machine learning model back then, which is a GRU, and we just added, like as a first proof of principle, we added an event generating mechanism to make the GRU, which you see up here, and augmented with event generating mechanism so that it operates with events. And yeah, we also used events on the input. So what you see here is like a event camera input that basically records only pixels that change their color value. And you get these on and off events that you see. And if you roll this out over time, you see that these are like really a lot of events, can be up to millions of events for one of these gestures that are presented here. And what we found is that actually you can do this. So this works in practice. So you can augment these machine learning models with an event generating mechanisms. What you see on the bottom right is that then you get very sparse output, which is exactly what we want. This sparsity we can then exploit to increase the efficiency. And you see everything that is white here is essentially where the cells are turned off, and only the gray values are like the spikes of this machine learning model now. And also we found that especially on these event-based data sets, these models also work very well. So we got really state-of-the-art performance back then on these event data sets, but also like on more standard machine learning tasks. Then we implemented this also on a neuromorphic hardware, which is really optimized for this event-based processing. So we made this implementation on the Spinnaker 2 board, which is developed at the Technische Universität Dresden, and we could show that we can actually get the same performance on a like back then like more simple language modeling task, but with a significant saving of energy, so around 94% energy saving, if you implement this bio-inspired algorithm on a bio-inspired hardware, which was very encouraging. So we could get up 20% energy savings with this event-based machine learning algorithm. But what we were a bit concerned of, it was not really truly event-based, our implementation back then. So it's not really an event-based implementation, and I will show you on the next slide what is actually the problem that we had back then. And with the help of Gauss Center funding, we could now improve on that and make a event-based algorithm and also test them on larger scale tasks. So what was the problem with eGRU? So if you think about the problem, it's like essentially what you see on the input is just events, right? So you have event channels. I show here 4, but there are really many in practice. And then what eGRU actually does is it chunks these events into frames, and then it does essentially a frame-based processing. So in every time step, you would only see kind of a subset of these events, of course, but still you would see them in a frame-based manner. But to utilize the full power of event-based processing, you actually want an what we call an address event representation, so that basically every of these events is just encoded by a certain encoding feature vector, and you process essentially one event at a time. So we worked basically since we published EGRU, we worked on this problem, and this is actually hard problem because, as I said before, this can be millions of events. So if you think of this gesture, which is still a relatively simple stimulus, it lasts maybe a second or so, but there are really millions of events. So you have essentially to keep track of all these subtle correlations between events that can be spatially kind of dislocated and also spread out over time. So how do we capture these long sequences? And over the last 2 years, we developed a model that we called Stream, that is able to handle these large number of events and do truly event-based processing. And the idea, I will just walk you through very quickly, is that if you have now an event stream as we have it in these DVS gesture videos, if you roll it out over time, essentially have these on and off events over the spatial XY coordinates. But you can always think of this as a stream of events over just the time dimension, right? They might come in very fast succession, but in the end, it's always an event stream. And we were looking for models that are actually able to work with such event streams. And what we resorted to in the end is a state space model. So essentially a simple linear recurrent neural network that is able to, or that was known already back then, to be able to learn temporal dependencies in very long sequences. And also it can be computed very efficiently. So it kind of gets the best of both worlds of recurrent neural networks. And we observed that it's also very well suited for event-based processing if you exploit the mathematics of it, which I will not go into details. So essentially we have then a model that looks like this. So we get these event streams and we have feature encodings of each of these events, and the model is able to really operate forward in time on this event stream. And we worked now with this for quite a while, and actually you can get these models to really achieve state-of-the-art performance, again, just by doing purely event-based processing. So we were the first to actually solve this task, this DVS gesture recognition dataset. To get 100% accuracy on them. And also we got on other more complicated tasks, we got state-of-the-art performance and could even outperform related approaches with purely event-based. So no frames, no convolutions, and millions of events per sample. Okay, I would go through a next quickly topic which we did more recently, which is a second principle that we observe in the brain. So brains obviously don't have clocks that would synchronize them across the whole brain. So they work intrinsically asynchronously. And this is a second principle we have been looking into recently. So what is the problem here? One has to go maybe a bit deeper. If you look on what essentially machine learning is today, it's using an algorithm that is called stochastic gradient descent. And this is applied to deep neural networks. And I made here like a cartoon of what's actually going on in SGD. So if you have a network with multiple layers, here it's 3 layers, L1, L2, L3. The algorithm essentially consists of a forward and a backward pass. And because these models get larger and larger, you typically distribute them over many, many devices. So we have here 3 devices, 3 layers per network. And we roll them out data distributed. So each of the devices would process a different chunk of the training data set. And we do then forward and backward passes through the network. And actually what we found experimentally is that the backward pass takes about twice as much time as the forward pass. And this makes also sense if you look into the math of what's going on. So this is a rule of thumb that you have around 1 to 2 imbalance in the algorithm. And then once the forward and backward are done, you have a synchronization barrier. So all of these updates that are computed during this time have to be communicated to all other devices. So you have to synchronize and then you have to communicate. So you have also an imbalance in terms of compute and memory access in the algorithm. So you have phases where there's a lot of compute needed, which is the forward and backward passes, and then where a lot of I/O is done. So we thought this is suboptimal, and we were essentially trying to get rid of the synchronization barrier. And I will not go into too much detail. So we also developed a theory that is based on kind of brain principles, but I will just jump forward to our solution. So essentially what we did is we tried to remove this imbalance by essentially just lagging these devices against each other so that they basically always are in different phases of the algorithm. And then we did fully asynchronous communication between these devices. So whenever one layer is ready, so to say, with new updates, these updates get communicated, and they're also not getting communicated in a broadcast as in normal stochastic gradient descent, but they are just communicated to one of the neighbors. So it's a local communication, which is also like a biological principle in the brain. So this is now an algorithm that fully removes a synchronization barrier. It does memory I/O and interleaved compute. And we have a theory that it works, and also in simulations we could show that this actually performs as good across a large subsection of what is done in AI. So I pulled here out a few results and we are working on more. So this is Cy400, this is like a standard image processing data set, and you see that in comparison to stochastic gradient descent and data distributed parallel, which is kind of the distributed equivalent of it, we get close to or reach state-of-the-art performance, but we reach the performance goal much faster, essentially. So we get speed ups of roughly 10 to 20% across all these tasks just by removing the synchronization barriers. The other one is ImageNet, and then also we are now working towards language modeling, and we see this consistently that these models actually scale well and also get these non-trivial speed ups. Okay, and that's basically what I wanted to show you. So in summary, I wanted to give you kind of a quick cross-section of what we're doing. So we're these two principles. So one is this EGRU model and stream, which we have been working on now, which allows us to process events in an efficient way, so up to millions of events, and also this asynchronous stochastic gradient descent, which allows us to decouple existing machine learning algorithms and to execute them more efficiently, but reach the same training accuracy. I want to thank my group and my many collaborators across Europe, and yeah, more collaborators and funding. And thank you very much.
Thank you very much, David. It was a very nice intersection and inclusion of this neuroscience insight into the new developments of artificial intelligence. Thank you so much. And then I think we can move quickly to Marvin, who I assume you will give the presentation, the last presentation. And then afterwards we move to the general quick questions and answers. So Marvin is working at the University of Darmstadt, and I leave the floor to you, Marvin.
Yes, thank you, Sandra. First of all, do you see the correct screen?
Yes.
Perfect. Okay, perfect. Yes, so I want to talk to you today about how we can utilize MPI neighborhood communication in large-scale spiking neural networks to improve the performance. And first of all, what is the problem? We want to have very large simulations. The brain has about 86 billion neurons and 100 trillion synapses. And simulating all of them or simulating a large amount of them is quite challenging. Because what do you need to do? At the end, you need to split all of your neurons on different compute nodes or different MPI ranks, and they need to communicate with each other. How is this a problem? So, of course, the neurons or the ranks can't do the work without communicating, because you have a lot of connected neurons over a lot of synapses. You can see an example here, and every spike that a neuron produces must be transmitted to the other MPI rank or to the other compute node. And now imagine this example that you see here with already a lot of errors, with 10,000 synapses per neurons, more neurons per rank, and a lot more compute nodes to simulate these 86 billion neurons. And you can already imagine how badly the communication will scale in such a large network. And let's take a deeper look at the problem. So neurons are connected with synapses, right? And synapses in the brain have a synaptic delay. And the good thing now for our approach is that we have the delay determines how much time we have to transmit the spike to the other compute rank. So in this case, we would have about 5 milliseconds in simulation time to transmit the spike from the source rank to the target rank. And if we have a randomly distributed network, like we can see here, we have basically random synapses between neurons and basically random synaptic delays. And at the end, what will result in that is that we have, for example, here, this compute node needs to send updates every 1 millisecond. This pair of nodes has 8 milliseconds, and it's there's no order. It's basically random how often the different ranks need to communicate with each other. And the easiest way to solve this in a simulation is we transmit all of the spikes at every time step using MPI altogether, but that scales very badly. So luckily, the synaptic delays in biology correlate with the synaptic length. And that's why we can now introduce our neurons into an Euclidean space. So we have a 3D simulation domain, and every neuron has a position in this 3D space. And we can see here now that neurons that are more close to each other have a smaller synaptic delay than neurons that are very distant from each other. And again, we can't communicate everything on this on one compute node. We have to distribute this on multiple compute nodes. So we can now partition our Euclidean simulation domain into smaller subdomains, and each subdomain is managed by a single MPI rank. And now, of course, we can transmit the spikes more easily, because you can see here an example. So one rectangle here in this graphic is one compute node, and the synaptic delays within a compute node are the smallest one, because the neurons within the compute nodes are, of course, very close to each other. So we need to transmit the spikes within the compute node, for example, every half millisecond. Then the next higher synaptic delays from this centered simulation subdomain are the neighbors of this domain. So for example, we need to transmit to our neighbors every 1 millisecond. And now we can do this with these MPI neighborhood collectives. So MPI neighborhood collectives basically transmit directly to the neighbors of our simulation domain. So there's no global communication needed, but the data is transmitted directly here from our source node in the center to the neighboring subdomains. And All of our ranks or all of our subdomains are doing this at the same time, but they don't communicate globally, only with their direct neighbors. And of course, we can do this now hierarchically. So we can not only use MPI neighborhood communication to communicate with our direct neighbors, we can also go one level further and communicate with the neighbors of our neighbors, for example, every 2 milliseconds. We can, of course, now go on and talk to the neighbors of our neighbors of our neighbors every 3 milliseconds and so on. But at some point, you want to stop to use this neighborhood communication because you will have some connections that are going to subdomains that are very far, and you have very few connections to this far domain. So it doesn't make sense to use the MPI neighborhood communication all the way. And at this point, you would use some kind of global communication. So for example, you could transmit here now using MPI altogether or MPI all-to-all. And yeah, so this is our approach. Now we can take a quick look at the evaluation. So we evaluated this on joules of the JC. And this is a weak scaling experiment, so the number of neurons per rank stays the same, but we increase the number of ranks. And yes, you can see that on the x-axis, and on the y-axis you see our simulation time. And basically, first we can look at the blue line, which is the altogether implementation, so a default implementation, and you can already see it scales quite badly for increasing number of ranks. An improvement would be using all-to-all instead of MPI altogether, which already reduces the simulation time by a lot. And finally, the 2 lines at the bottom are our neighborhood implementation. And we can see that we can reduce the simulation time by almost a factor of 4. We have here 2 different lines because first we use neighborhood combined with altogether and for the global communication, and the other line is for all-to-all. And we can also see that all-to-all there is a little bit better than using altogether in this case. But yeah, we could improve by almost a factor of 4 by introducing this neighborhood communication in the Euclidean space compared to the standard altogether implementation. That is probably the easiest one. So yeah, let's sum up our results. So the communication was the bottleneck of these large-scale simulations. And in our approach, we exploited the spatial locality and utilized the MPI neighborhood communication to reduce the communication overhead. We were quite successful for that. We improved the performance by a factor of 3.7. And we believe that larger networks would benefit even more from our approach, because you can already see here in this graph how MPI altogether scales with increasing number of ranks, and it scales quite badly. And our approach is not constant, but it scales, it increases relatively small compared to the increasement in simulation time in altogether. So yeah, we think larger simulations would benefit even more from this approach. And yeah, with that, thank you for the opportunity to speak to you today. And also, I'm happy to answer any questions later on.
So before we jump quickly into the Q&A, I was asked to share with you an invitation to join the eBrings Summit that will take place in Brussels between the 8th and the 11th of December. If you are interested to join, there will be different opportunities to network, to participate in the open day, and also to participate in the plenary presentations and the scientific conference. I leave here for you as well a link to the registration. If you are interested to join us, I think that would be very nice, and we would be delighted to see you there.